It's 8 o'clock, so let's get started. Okay, so welcome everyone to our new Malaysian Research Insider session in 2021. I hope everybody is staying safe and healthy. To those who are new to the Research Insider session, allow me to quickly brief you about who we are and what this session is. So this live stream will be streamed on the MBIOS Facebook page, providing a chance for everybody to discover our talented Malaysian bioscience researchers and also a platform to interact with them. Each scientist will discuss their work, their experiences in science, and how they got to where they are now. These conversations will be accessible to all and are directed at non-specialists. The general format is that the speaker starts with an overview of themselves and their topic of conversation for the day, followed by a Q&A session. If you are interested in science or learning about topics outside of your area of expertise, please don't forget to tune in every third Saturday of the month, prepared to chat with a Malaysian scientist and to learn something new. I'll quickly introduce myself. I'm Ashwini Ilangovan, a PhD student in biomedical sciences at the University of Pittsburgh. This science chat is organized by the Malaysian Bioscience Scholars, MBIOS, and media partnered by 100 Scientists of Malaysia and Science Media Center Malaysia. A quick reminder for the audience, please feel free to post any questions in the chat box at any point during the talk. All right, so our speaker tonight is Menusha Arumugam. Menusha is currently a PhD candidate in molecular and cell biology at the Andel Institute in Michigan, USA. Her research focuses on studying how the tumor microenvironment, specifically adipocytes and collagen, can contribute to tumorigenesis in NF1 deficient breast cancer. Without further ado, let's welcome our speaker for tonight, Menusha. It's all over to you, Menusha.
Thanks, Ashwini. Thanks for the nice introduction. So I wanted to start my introduction slide with these images because, well, one, it's really pretty, but also these are the two methods I'm going to talk about my research. So yeah, I hope you stay tuned towards the research I'll be talking about is towards the end. So I hope you'll stay tuned towards the end to hear about it more. Okay, so I wanted to start by talking about my educational journey to where I got to where I am today. So all these places have really contributed to shaping me as a student and as a scientist. Without all the people I've met at these places, I wouldn't be where I am at today. So I started with going to SK and SMK Common Kajang. So I did my standard 1 through standard 6 there, as well as Form 1 to Form 3. And after PMR, I applied and got accepted to the Permata Pinta program. And this was a program in, the campus is in UKM. So at Permata Pinta, I did my Form 4 and Form 5. And during my Form 4 and Form 5 time there, I was exposed to research. And this is really where I started to think about science and research as a potential career. Before that, I really wanted to be a doctor. And I'll talk a little bit about my experience shadowing a doctor as well as doing research in Permata Pinta. So after Permata Pinta, I applied to the Asasi Pinta program that's in Permata Pinta as well. And that was maybe, I think, a 10-month, 8-month program before going to UCAM. So then after Asasi Pinta, I started my degree at UCAM. So during my time at UKM, I also got accepted for the My Brain Science Scholarship, and they gave me the opportunity to apply either to an overseas university or a local university. And depending on which one I choose to go to, they will fund that. So during my first semester, after I started at UKM, I got accepted to University of Michigan Flint. So then after I finished my first semester, I moved to University of Michigan Flint in April of 2017 and continued my degree there. So I finished my degree in about 3 years because of my 1 semester here and the Asasi Pintar classes that I took. Those credits were able to be transferred to University of Michigan, and I graduated in 2017. So during my time here, I was in the honors program. And the honors program had the opportunity for students to travel to a different university to do an internship program. And this is how I did my internship at Columbia University. So my time at Columbia University was actually what solidified my decision to go into science as a career and pursue a PhD. And I'll talk a little bit about what research I did in Columbia as well. And then after Columbia University, well, this was in the middle of my undergrad career. So after I graduated from University of Michigan Flint, I'm now here at Andel Institute, where I'm doing my PhD in cell molecular biology. So at UCAM, well, at Pamata, all students were given the opportunity to shadow a mentor in the field of your choosing. So at that time, I chose the field psychiatry because that was what I was really interested in. I wanted to go to med school. I wanted to go into the psychiatry field. So I shadowed a doctor at UCAM, and I asked him about research, like what research can I do? Because we're all required to finish a thesis project in Form 4 and Form 5. So while we were talking about research, this is what we came up with. We wanted to study what patients thought about electroconvulsive therapy for depression. And electroconvulsive therapy is basically, so they place these little electrodes on the surface of the patient's brain and give short electrical currents. And it creates an environment that's kind of like a small seizure, and that sort of improves the brain chemistry for patients with mental health issues. So overall, this is a really good therapy, but patients tend to be scared of it because of how it's displayed in movies and things like that. So this is one of the research I did with my mentor. While this was great research and my experience shadowing him was really good, he was a great mentor, I feel like this wasn't really a good fit for me. I wanted something more molecular, something that I can go more into the nitty-gritty of the biology. So then that's really when I decided to really give up on the med school dream and pursue a degree in general biology. So at UVM Flint, so none of these pictures are science-related pictures, but I wanted to bring this up because this is really important in your experience as a student. I feel like as much as I love science, I loved all the things I did outside of science as well. I was very involved on campus. I was involved in student government. I was an international student ambassador that helped international students get adjusted to American university life. I went to leadership seminars, and I made friends that I'm still friends with after graduation. So as I mentioned before, while I was at U of M Flint, I was in the honors program, and they gave $3,000 for every student in the honors program to do an internship. So I chose to do my internship at Columbia University. And this process was actually quite, it's an interesting process. So there wasn't a streamlined process for us to be accepted into an internship program. You had to cold call professors around in whichever university you wanted to go to. So I wanted the experience of living in New York, and Columbia had a really good biology program, and I was very interested in this lab. So I emailed the PI and like many other PIs in labs all over the US, and he emailed me back and said, sure, come to my lab. You can spend 10 weeks here learning with one of the scientists. So at Columbia, my project mainly used this model called C. elegans. And I'm going to ask for my first poll question to be released now. There we go. All right, we'll give a couple seconds. So how long do you think an adult C. elegans? So this is what the adult C. elegans look like. All right, so it looks like most of you answered 1 millimeter. That is actually the correct answer. Good job. So yeah, so in the adult stage, it is 1 millimeter long. So just give me a second. There we go. It's 1 millimeter long. And this model is really important to the question that we're studying because of this downward stage. So this downward stage is really a stage that the worm can go into when they're under stress. So for example, so this is the eggs, and then as it grows up, if at any of this stage we choose not to give them food or put them in a temperature that they don't like, they can turn into this downward stage where a lot of their body functions is limited. So they don't use as much energy. They're saving themselves. And then if you give them food back again, they'll go back and continue their life cycle just like normal. So the question that we were studying is, if you starve a worm at any of this stage, does the babies of the next life cycle, is there something different about their genetic? Can they respond to the stress better or at the same level of their parents? So the project I was doing was really looking at the physiological side of it. So whether these worms move differently, move faster, turn a different way. And the mentor that I was working with, she was studying the more genetic side of it. So this was a really interesting project, but the sad story of this project is that at some point, the plates that we were growing these worms on got contaminated. So everything that I was studying for the past 10 weeks is not valid anymore. So we had to throw away all the plates. So it's kind of disappointing, but that really gave me a reality into what science is like, because now in my PhD, there's a lot of failed experiments. And what gave me a sign really to that this is what I want to do is that even though those experiments failed, it didn't upset me. It didn't steer me away from the field. And this is really a sign that this is what I want to do. I want to do biology research. Right, so this is where I'm at now in Grand Rapids. And I wanted to bring up this picture because this is really one of the important reasons that I chose to go to Van Andel Institute. So this is Van Andel Institute. And this street over here is what they call the medical mile. And the reason why they call that is because within 1 mile, there's a lot of medical institutions. So there's Van Andel Institute, a biomedical research company, research institute. There's Michigan State College of Human Medicine, and all these buildings over here are hospitals. There's a Butterworth Hospital, there's a Children's Hospital, there's a Heart Center. And all these institutions, including Van Andel Institute, have a very good relationship with each other. So, for example, in my first year, and even now, we have the opportunity of attending what they call tumor boards. So hospitals, at least these hospitals, I don't know if other hospitals do it too, I'm sure they do, but they have what they call tumor boards, which is basically a meeting they have, I don't know, depending once a week, depending on their department. So all the doctors, radiologists, pathologists will gather for this meeting to discuss some of their most difficult cases. So typically in one of those meetings, they go through maybe 5 cases, depending on how complicated they are. But us as students, we can see how these different roles in the hospital discuss and talk to each other about a certain disease. And that's really the type of research I want to be involved in. Research that's very translational. So everything that you discover in the bench can be applied. Almost everything that you discover in the bench can be applied to the clinic, or that's the goal of your research, being able to apply what you learn to help patients. And being in this environment has really helped me grow as a scientist to think about research in a translational perspective. So I also just wanted to go through like what my life is like in grad school from when I started to now. And so what I'm talking about today might be very specific to Van Andel Institute. Other universities might do it very differently. So I'm going to ask for the second poll question to be shared. Right, so what do you think is the average PhD time to graduate in the US? I don't know if you can see the whole question, but. All right, so it looks like most of you answered 4.5 years. So the correct answer is actually 5.8 years. So based on what I Googled really quickly, when I was preparing for this talk, is 5.8 years. And this was data that they collected in 2017, I think. So maybe that has changed recently. But on paper, PhD programs in the US is a 5-year program. When you get accepted, they tell you it's a 5-year program. But usually it can go longer than that. And I'll explain why it's sort of like a flexible end date. So during your first year, so you can come in either with your master's or your undergrad degree, and I came in with my undergrad degree. And so in your first years, you'll be taking core classes, or at least this is how my graduate school did it. So I took core classes, so classes that are required for me to graduate. And I also did rotations. So what rotations are is that you get to be in a lab that you might be interested to join. So this is sort of you just surveying the lab. So I was in 3 different labs for 7 weeks each, just learning what the lab environment is like and what the mentor is like. So during your times in rotations, people advise you to choose your mentor instead of choosing the science, because usually you can learn to love a lot of the science that you're in. Of course, don't pick something you absolutely don't like, but the reason why choosing your mentor is very important is because they're the ones that's going to train you. Whatever you do in your PhD, like what research you do, is not necessarily the same science you have to stick with for the rest of your career. In your PhD, they're just teaching you how to do science. So to learn how to do science, you want a mentor that can match your personality style and the mentor that you need. So I came in with only 10 weeks of lab experience. So I wanted a mentor that would be okay with teaching me everything from scratch. So that was the mentor I was looking for, and that was the mentor I found. So this is a picture of me and my cohort during our first year. This was first day of grad school. We had an event to welcome us to the grad school. So another important thing that we have during our first year is our thesis advisory committee meeting. So after you get accepted to your lab, you have this meeting to sort of help you plan where the direction of your research is going. And this is a group of maybe 4 faculty members that will help you throughout your time in your grad school. So in my second year, I had elective classes. So I can choose from a selection of classes that they offer, but I still need to fulfill these credits. And then one of the big things in my second year is my comprehensive exams. So the comprehensive exams is basically you proposing what experiments you want to do for the rest of your PhD time. And then they will grill you on your proposal. So they'll ask you questions. What if your experiment fails? What are you going to do? What if your data doesn't show this? What does it mean for what does that mean for your question? Are you answering the right questions with these experiments? So it's a pretty intense exam. It's about 2.5 hours long exam. It's an oral presentation. So it's pretty difficult. And this is me and my classmates after we all passed our comprehensive exams. So once you pass your comprehensive exam, you're officially a PhD candidate. And then you also have your TAC meeting. And as you go on, you'll have your TAC meeting twice a year. Depending on how many classes you've taken, you'll still be taking classes, and you'll also be focusing on publishing papers. So this is where I'm at. I'm at the end of my 4th year, in finishing up my 4th year in August, September, I'll be starting my 5th year. And then in your 5th year, you have your dissertation defense, where you defend everything, what you've proposed and all the experiments that you've done for you to graduate. So this is my lab. And these are to my 2 mentors. So officially, the lab is called the Steensma Lab, and he's an MD. So do you remember that picture I showed you, the medical mile, all the hospitals around Andel Institute? So he's actually hired in one of the hospitals that's across the street from us. And his role in the clinic is very important to maintain that translational research environment that we have in the lab. Dr. Carrie Graville, she co-mentors me. She also runs the lab with Dr. Matt Thiensma, and she has a PhD. So she brings a lot of the research, molecular biology side of research into the lab. And both their personalities and both their scientific expertise really balances out the lab really well. And these are the other people in my lab. They're graduate students, they're tech, they're postdocs. We also have a clinical fellow. She's a doctor, so she's an oncologist across the street at the hospital. And part of her fellowship, she has to do a research year. So she also brings a clinical perspective to the environment in the lab. So the research that we study in the lab is called neurofibromatosis type 1. And this is a genetic disease that's usually diagnosed based on the symptoms. Different patients can have many different symptoms. So for example, it's lymph nodules, this little red spots in the eye, some cafe au lait spots, so this discoloration on the skin folding and freckling. And one of the ones that we study a lot in the lab is what we call neurofibromas. So it's little tumors on nerve cells. And these tumors can form either on the skin or under the skin. And we study this a lot because of also Matt's role in the clinic. So Dr. Steedsma is able to take samples from patients in the hospital and bring it to the lab for us to study. Right. So if you want to learn more about neurofibromatosis type 1 or NF1, I really recommend Googling this twin. So you can Google the Pearson twins, and they have a short documentary on YouTube about them. So they are basically identical twins. So everything about their genetics should be the same, or is same. But the way NF1 works, or because we don't know a lot about NF1, we don't understand why, even though they're identical twins, the way they manifest the disease is very different. So one twin has really a lot of tumors on his face, and this twin has no physical manifestations, but he has a lot of neurological issues. So he has memory issues, and he also was diagnosed with epilepsy. So if you have time, it's about an 11-minute documentary, and if you're interested to know more, definitely Google it and watch it. Super interesting. So one of the cancers that an NF1 patient can get is breast cancer. And in breast cancer, so usually the cells so this is what your breast would look like normal. So this is the gland, this is where milk will be produced. So different cell types that make up the gland, and there are different cell types that's around your mammary gland. But in cancer, the cells that are your mammary gland is what becomes the cancer. And it starts to migrate out into the other parts of your breast. So the cells around it can actually also contribute to how aggressive the tumor is. And all the cells and all the components around the tumor is what's called the tumor microenvironment. And now I'm going to ask for the third poll question to be released. So I mentioned the tumor microenvironment. What do you think is part of the tumor microenvironment? Is it immune cells, fat cells, fibroblasts, collagen, or all the cells? Right, so it looks like most of you answered fibroblasts. That is correct, but all the other answers are also correct. So the correct answer is all of the above. So in this figure over here, what I'm showing you is the mammary gland. So that's epithelial cells, and the cells around it. There are fat cells, there are immune cells, these little purple ones. There are fibroblasts. So fibroblasts are cells that spit out different stuff to make your connective tissues. For example, they produce collagen. collagen is also part of your tumor microenvironment. And the reason why studying the tumor microenvironment is so important is because while the tumor undergoes like changes, the cells around the tumor that are not the tumor also changes to help the tumor become more aggressive. So like over here, I'm showing you the fibroblasts are skinny, and over here, and in cancer, it becomes large, it migrates, it can move a lot more, it can spit out a lot more things that helps the tumor survive. So, and moving forward, so what I'm going to talk to you about is how to study the tumor cells on its own and how I'm studying the tumor microenvironment as well. Right, so this was the figure that I have from my title slide, and basically what I'm showing you is 3D culture immunofluorescence. So immunofluorescence is just a way to measure different components that you're interested in the cell using antibodies. So that's why they call it immuno, because it's using antibodies, and fluorescence just means emitting light. So there are two different types of way you can grow epithelial cells or cancer cells or any type of cells. You can grow them in 2D, which means it's flat on a plastic plate, or you can grow them in 3D. So a lot of the work has been done in 2D culture, so flat on plastic plates. So we've learned a lot by studying cells on 2D, but we might be missing something in biology because this is not how our cells are in our body. One, it doesn't touch plastic. Cells in our body doesn't see plastic. And also it's not flat. And it has other type of cells and other type of things around it. So in 3D, we're growing cells for it to become in a more physiologically relevant environment. And to help you understand what I'm saying is that, so I have this figure. So these are breast mammary gland cells. So if you look at them, they're forming this really circle shape, just like the figure I showed you before. So these are how normal epithelial cells look like. They have this little lumen in the center, and this one also has lumen. But when you mutate these cells to become cancer, they start forming this unorganized structure, just like how cancer would. So this is why 3D culture is really important. The cells get to grow in a form that they would in the human body. In 2D, you miss all these important changes because they're flat. And so this is what I'm trying to study, the NF1 breast cancer. This is how I'm trying to study the NF1 breast cancer cells in my project. So this is an example of the cells that I've grown. So the top 3 images are normal cells, so non-cancer cells. These are just looking for different, staining for different markers. This is the nucleus, and this is a type of protein that you find around the nucleus or outside of the nucleus. And this is what, when I mutate NF1, this is what the cells look like. They're no longer circle, they no longer have the lumen in the center, just like a normal mammary gland would look like. This becomes more really just very unorganized, irregular shaped. So this is one way that I'm studying the tumor cells. So this is the other figure that I had on my title slide. And so this has a longer name. It's called second harmonic generation imaging. There's a lot of physics involved in this imaging method, and I won't go into that today because I'm not the best person to go into the physics details. But basically, the 3D structure of collagen allows it to be imaged on this image, and it's those are what you see in the white sort of wavy type of wavy type of structures on this image. And what you see over here, these are what your mammary gland looks like. All right, so why do I want to study collagen? So we have a rat model in the lab that it has NF1 mutation and develops breast cancer. So when the rat doesn't have the NF1 mutation, so it's normal, not developing cancer, this is what the mammary ducts look like. So this is the ducts, this is the fat cells around it that I talked about. But when we mutate NF1, we see that around the ducts, there are this like pink sort of things. But in this image, the way we're taking this image, we don't really know what these pink things are. But what we think is that it could be collagen. And it's really interesting. And why collagen is because collagen is actually the most abundant protein in your body. And we're really interested to look at this because collagen can actually, the more collagen is found around the mammary gland, you have an increased risk of developing breast cancer. So using second harmonic generation imaging that I talked about earlier. So these images are not from my research. I actually forgot to put the paper that I took this from. But so these are second harmonic generation imaging. And over here, they're taking an image from a small tumor. So this is a very small tumor, and this is what the collagen looks like. They call it curly. So sort of wavy, curly, not very organized. But when they take an image of a large tumor, so this area is where the tumor would be, the collagen starts to become straight. And what's important is between a large and invasive. So invasive meaning it starts to like travel out of where the tumor starts to grow. You can see that the direction of the collagen is changing. So over here, you can see it's like parallel to the tumor boundary, but over here it becomes perpendicular. So papers have described it as sort of like a highway for tumor cells to migrate out of the tumor. And you also notice that there's generally a lot more collagen here. So with my project, I really want to look at not only if with NF1 mutation is the amount of collagen increased, but also is the direction of collagen changed in NF1 patient and the NF1 rat model that we have in the lab. Right. So what am I doing with all these science, right? Like where's all these science going? So one of the most exciting things I think about being in science is being able to share your research and being able to learn your research at conferences. So at Van Andel, they provide $2,000 for graduate students to travel to conferences, and I've gone to a conference every year, well, except this year, I guess. So in 2018, my first year, I went to Cold Spring Harbor conference where I met, so these are students that I met over there, and I was a first year graduate student at that point, so I didn't have a lot of data to present, but this was a really cool place to visit because a lot of really important people in science are actually working at Cold Spring Harbor. And some friends that I make, I also meet the following years at different conferences. So this is a different conference I went to in 2019. This was a very specific mammary gland meeting where all the researchers talked about breast cancer only. So my first meeting was just general cancer. And then I started focusing on breast cancer. And these are my mentor, and usually the meals at these conferences are really fancy. And it's a lot of fun. So my second year, I had more data to present a poster. And then my third year, last year, I actually traveled to Portugal for a conference looking at specifically the tumor microenvironment. So I also presented a poster, and this was actually the view from outside the conference center. So it's a lot of fun. And so I mentioned that I made friends. So she goes to Michigan State University, so it's like an hour away. And in our daily lives, the chances of us meeting each other is very low. But at this conference, we met each other. We're still friends until today. We talk about science all the time. And you also meet a lot of different scientists, different faculty members from different universities that become helpful for your project now. One of the faculty I met in my first meeting is on my thesis advisory committee now, and he's helping me with my experiments for me to graduate. So I wanted to end with this slide because this is one of the most rewarding experience that I had in my graduate program. So my lab, because of the community that we have in Grand Rapids and my mentor's role in the hospital, we maintain a very strong relationship with local patient advocacy groups. So the pictures from the top is from a team called Be Brave. And they're a group of women from the Grand Rapids area. And most of them are actually breast cancer survivors who put together this fundraising event called Be Brave 5K. So every year in October, they organize this 5K, and they also have a golfing fundraising event. And all the money that they raise goes to our lab. And this event is so I'm not a runner, really. I run a little bit. And I'm usually not very happy when I'm running. I'm usually sort of suffering when I'm running. But at the beginning of this event, they have survivors of breast cancer speak and talk about their story. And it's very inspiring to hear these women talk about their science, talk about how they've overcome what they've gone through. And they've also visited our lab throughout the year and asked us what we're doing in science. And that experience really trains you to learn how to talk about your science in a way that the public can understand. And the pictures down here is from this organization called NF Michigan. So they're a neurofibromatosis patient advocacy group that also fundraise and donates money to our lab for research. So meeting all these patients and meeting all the survivors really gives me motivation, a sense of urgency, and perspective of what I'm doing. So even though a lot of my experiments failed and research can seem really slow, you're really reminded why you're doing what you're doing, and those things are important, even with all the failed experiments. So yeah, that's all I have for today. I hope I gave enough time for questions. Do you want me to keep sharing my slides, or?
I think, yeah, I don't think we need your slides.
Okay.
Okay. Thank you so much, Menusha. That was a really nice talk all about your journey, as well as a little bit about your science. That was really nice. Thank you again. Okay, so we'll start with some general questions. How do you keep your motivation going while you're, in this PhD journey, this very long PhD journey?
Yeah, so I think the first thing is that I definitely love what I do. I was listening to a podcast the other day that said that a lot of the stress that we get from work, if you actually like what you do, a lot of the stress that you get is from outside of work. So, and when I thought about it, that's true. When I'm in the lab, when I'm reading about science, or when I'm doing my experiments, I'm not actually stressed. I actually love what I do. It's fun. A lot of my stress comes from like outside of the lab. So when I'm done with my experiments for the day, I go home, or like if I'm done with experiments early and I go home, for example, like 2 o'clock, if I'm sitting at home and I'm like, should I be at home at 2 o'clock? Should I be in the lab until like 6 o'clock? There's all this stress worrying about, am I doing enough? So one of the things that really helped me is to just accept, yes, I am doing enough. There's always more experiments that I can do, but do I need to do them? Will I be okay if I do all the experiments in the world, So just really taking time to take care of myself has kept my motivation going. If I worked, 80 hours a week, something crazy, I will not have the motivation anymore. Another thing that really helps me is my lab environment. So my mentors are very supportive. I'm actually using their office right now for this presentation. They let us use their office whenever we have presentations or we need a private space for anything. So they're very understanding that we have lives outside of our labs. They really make that very important. You have your families, take vacation time, don't get burned out. And they try to maintain an environment where we're very collaborative. We started out as coworkers, but now we're all friends. And we can always go to each other for questions in science, or even, they help me stay motivated. If experiments are failing, they remind me it's not me. I'm not a bad scientist. It's just sometimes experiments don't like you, But yeah, that's how I've been maintaining my motivation in science.
I cannot agree more, especially the first part. Yeah, it is really hard to know when to stop, and it's really easy to start doubting if you're doing enough. Yeah. It's hard. Mm-hmm. Okay, so what do you find to be the most rewarding in this journey of yours?
Yeah, I definitely think it's my last slide where I talked about meeting patients and going to fundraising events. That's definitely has been the most rewarding experience for me. I never thought about how that could be a part of my PhD education. I always thought it's going to be lab and classes. But because of the way my lab is set up and because of who my PIs are, they're also very involved in the community. That experience has been really rewarding to me because it reminds me of what the big picture is. When you're doing experiments, like when I was talking about, these collagen imaging and the immunofluorescence, it gets into the very nitty gritty details. But then when you meet these patients, hear their stories, and you go for these fundraising events, it reminds you of the big picture of what you're doing. Someone's waiting out there for you to discover what you're discovering. Even though everything can seem like it's going slow, research is a slow process. Something that I find might not go into the research right away, but it'll help someone else discover what they want to discover. And there are patients out there waiting for this. So I think that has been the most rewarding experience. And I actually really enjoy meeting them when they come visit the lab. So they come visit the lab, and we have different stations in the lab where different graduate students and scientists will talk about what they're doing. And it's good experience talking about your science without the nitty-gritty details. Like, do you understand what you're doing, why you're doing what you're doing, in the big picture of your science? I think that has
Absolutely.
been
Yeah, the big picture definitely helps put you on track to realize what you're doing. And like you said, someone's waiting, and it can get a little bit scary at times because you are dealing with something huge, although it doesn't seem like it on a daily basis. But yes, knowing that you're doing something, a little part into it, it's very rewarding. I totally agree. Okay, so we have a question about whether COVID has impacted your research plans.
Yes, it has. So when COVID first started, we had a lockdown. So the institute closed. Different labs, different institutes sort of does it differently. But my mentors took it very seriously. They told us all to stay home, don't come into the lab. Only 1 or 2 people were allowed in the lab, and that was, just to take care of the rat models that we have. So a lot of the models that we had to be reduced in number. No one else was doing experiments. And I think I was maybe at home for a couple months. So yes, that definitely put a hit on my progress. But there was things that we were doing from home. So one of the initiatives that the hospitals across the street wanted to do was to create a database for all the COVID papers that was coming out. So the doctors in the hospital were struggling to keep up with the amount of papers that are coming out, and not all papers are really relevant to finding information like what treatments out there to help their patients. So they reached out to graduate students in Van Andel to see if, do you want to read these papers, and then fill out a form that ranks these papers. And then once we rank the paper go into a database that clinicians can quickly search depending on who they're looking for. So we rank, is this paper studying pediatric population? Is it studying patients with diabetes? And then doctors can go in and quickly search, I have a pediatric patient with diabetes. What are the papers out there that look at this? So that was what I was doing at home for COVID, but definitely research has been impacted. I'm fully back in the lab, but we can't have a lot of in-person meetings, or we can't have too many people in a room at the same time. So yeah, it's impacted.
Very nice. Okay, so what are your future plans in science, and how does that align with your major goals?
Okay. So my future plans in science. So as I mentioned, I chose Van Andel because of its close relationship to hospitals and its translational environment. So I know that's really the environment that I want to keep being in. So after my PhD, at least in the US is how they do it. After your PhD, you usually do a postdoctoral fellowship. And I haven't decided where exactly in the US that I want to do my postdoctoral fellowship, but I definitely know I want to stay in a lab that's very translational. So a lab that's attached to a hospital, or a university that works very closely to a teaching hospital. So that's a lab environment that I want to be in. And I think it still comes back to my goal, and comes back to the most rewarding experience I had in grad school, is meeting these patients, talking to patient advocacy groups, and knowing that some of these things that we find is going to the clinic and someone's waiting for that. So that's definitely what I want to do in the future.
Very nice. Okay, so we have a question from Nazmi on Facebook. What are the hardships that you have faced so far in your career, and how do you go about it? This is coming from a current undergraduate, and would love to have tips on what he can do.
Yeah, so I would say, trying to think, okay. So in undergrad, one of the hardest things I think was really trying to find a balance between doing the things that you like and doing the things that will get you into graduate school. So for example, I was very involved on campus in like non-science related things like student government, in the international center. So all those things it really developed me as a person, gave me a lot of confidence for public speaking, gave me leadership skills. But I did a lot of that, and it was kind of late when I realized I didn't have a lot of research experience and I'm going to need that to get into grad school. Um, so after my internship at Columbia University is when I started applying to graduate schools, and a lot of the graduate programs were rejecting me because I didn't have that much experience in research and I haven't presented any of my research in conferences. So that really was one of the big stressors in my life, I would say, in undergrad, because I had so much fun doing all these things that developed me as a person, but then it was not helping me really get into grad school as much. So I would say there wasn't really, I guess I didn't really overcome it myself. I still got accepted to grad school. But if I had an advice for an undergrad, try to balance out those experiences. Try to start it earlier on in your undergrad career. I started really seriously thinking about this in my third year of my undergrad. That was sort of kind of late. So if you have the chance to think about it earlier, get into research earlier, I would definitely encourage that. In grad school, I would say a lot of the hardships that I face is really just doubting myself. Like because science is a field with a lot of failed experiments, and it's not really a field where you see rewards a lot. I would say from like the image that I showed you of the 3D cells of the video, that took me 2.5 years to get that. The 2.5 years was just me trying to grow them. They're not growing, trying to stain for that protein. The antibodies didn't work. There's a lot of failures. And then the image I showed you, imaging the collagen, I just started that maybe a little bit over a year ago, and I'm still trying to get the right image. So it's a field with a lot of failures. And I think I maybe only hear like, you're doing a good job, like maybe twice a year. It's not that people are mean. That's just the way science is. And I think the way that I've overcome it is to not doubt yourself as much. Don't think you're a failure. I mean, I still think that sometimes, but don't take that to heart. Don't stop what you're doing because you think that. It's just your head telling yourself that you're not a failure. So, yeah.
Yeah. I don't know what to say. I can't agree anymore to that. It's really, really hard to lose track of what is actually the more important thing.
Right.
And at some point, you just start realizing that you take any little success that comes your way, you're going to take it, you're going to celebrate it because you never know when it's going to come again. Yep. But I mean, not to scare people off, this is a very rewarding path.
Yes.
It's
Yeah.
just, it's not easy, but everybody knows that PhD is not easy. It's not meant to be easy because you are discovering something new. And it comes with all these challenges, but at some point you're going to look back and think, wow, I did all of that. I didn't really just learn science. I learned also kind of about life too, and about myself. It's really a journey of discovery of a lot of things. Okay, so what would your advice be to someone who's thinking about pursuing a PhD?
Yeah, I would say definitely try to do an internship or do a short type of maybe work in a lab that does research. It doesn't have to be, you don't have to think too much about the type of research they're doing because that doesn't have to be what you're going to do for the rest of your life. Just really whichever lab that has the funds or has the ability to accept you as an intern. Spend some time there. See if that's something you want to do. Because that's really how I learned that I didn't want to go to med school and I want to go to research. I shadowed the doctor during my Form 4, Form 5 years. That's how I decided seeing patients wasn't really a fit for me. But then at that point, I still don't know if research was a fit for me, right? So in undergrad, my experience at Columbia University was really what, told me, yes, this is something I want to do. So I was in the lab. I loved going to the lab. Even when things failed, I still loved going to the lab. So I would say that's definitely what you should think about when you want to go into research. But a PhD specifically, I would say it's not, I would say it's more of a time commitment. That's 5 years, well, at least in the US, that's 5 years of your life that you have to commit to doing research. And it's also, you're not getting that much money. So you're not going to make a lot of money. So it's also going to be a financial hit. So if you have a family that's depending on your income, that might be something else you need to think about. So I would say definitely take some time to think about financially if that's something you want to do, time commitment wise, if 5 years that's something what you want to do. And sometimes you could still be in research by doing a master's program. You don't necessarily need a PhD to be in the master's program. But if you one day want to have your own lab, or if you're thinking about having your own lab, it's worth it going through the PhD program. And that is what I want to do. At some point, I do want to have my own lab. Haven't decided at what stage in my career I want to have my own lab, but yeah, for sure, that's something in the future for me.
It's definitely very refreshing to hear someone say that you do want to start up a lab, because academia is not something you hear too much anymore. Yeah. That is very nice. Okay. Oh, okay. Someone asked, I think there's a blurry line between molecular biology and genetics. How do you differ between the two? Or how do you distinguish between the two? I don't know how to answer that either.
Yeah. Well, I don't usually try to differentiate both of them, because I think it's usually split up in the context of like classes and when you're learning things. Like in university, you learn molecular biology and then you learn genetics. But really both of those things overlap a lot when you're in like in the bench, or when you're trying to publish papers, because you can't have one without the other. For example, like the cells that I showed you, the 3D cells that I showed you, we're trying to publish the phenotype that we're seeing, the molecular phenotype that we're seeing, but we can't publish it without explaining the genetics that happen. Like what mutations do we have, and what does that mutation mean to the cell? Both of those sort of go hand in hand. I don't know if I'm 100% answering your question, but I think in like the bench science and the science publishing world, both of those fields really overlap. And I think the blurry lines are there for a reason. You can't have one without the other, really. Does that answer your question?
I think so. I just feel like molecular biology is like the bigger umbrella term for many things, and probably genetics would be one of that too under it. Okay, so I did see a few comments from your family, so I want to end with this question. Who is your inspiration or a role model, and who supports you in this amazing PhD journey?
My family asked me that?
No, your family just commented the congratulations, but yeah. I'm sure they are part of who you are and how well you do. So yeah.
My role model. At this point, I think I've had a lot of different role models throughout growing up, like in undergrad, in Pamata and Kanmukajang. But the role model I think of right now is my mentor. So Dr. Kira Graville that I mentioned, she is a great scientist, but she's also a great person, and that's really what I aspire to be. Sometimes I think people forget to be personable as you get successful, but she's very humble. She once accepted an intern to work in our lab when she met them at the gym. Like they were working out together, and she was like, oh yeah, I'm in high school and I'm trying to decide if I want to go into research or medical school. And she was like, no, come work in my lab and see what you like about research. Just very humble. And she has a great work-life balance. She does CrossFit on the side, is a hockey player, has 2 kids, just living a great life. I feel like she's my mentor. She doesn't put aside the science to have a good family life, but she also doesn't put aside her family life to have a good son. She has found the balance, and she's just a great person overall. It's who I want to be when I grow up. But also, I wouldn't tell this in person because then she'll make fun of me.
Of
Model.
course.
So, yeah.
And yeah, who supports you in this journey?
Oh, who supports me? Yeah, definitely my family. There's been many times when my parents would be like, "Oh, can you call today?" It's like, "No, I'm in the lab," and they're very understanding about that. I want to travel to Portugal alone for a conference, and my mom was like, "Okay, sure, go ahead." you know. Yeah, my family's been very supportive. Definitely my number one cheerleaders. My friends that I meet in graduate school and undergrad, my friends throughout. So every stage of my career, we're still friends, no matter how far that we are. Some of my friends are in Malaysia, some are in Europe. We still keep in touch, and definitely motivate me whenever I think that I'm not doing enough or I'm not good enough. And they're like, "No, you know what you're doing. Keep going." Definitely my mentors, my coworkers in the lab, very supportive. And my friends that I meet in grad school, because we sort of started the program together. We all started, doubting each other. Do we belong here? And at each stage of our life, we celebrate each other. And I think that's been really nice. Like when we all pass our comprehensive exams, we celebrate each other. My roommate, she's also in the program with me. When she got a grant, we celebrated her getting a grant. So really, it's definitely, I don't know, it's really everyone in my life has been really supportive.
It's really nice to hear. Okay, I think that's all the time we have for today. So thank you very much, Menusha, for the very informative presentation today. And thank you for everybody else on the Zoom call and also on Facebook Live for tuning in. For those of you who joined late or missed our chat, you can watch the edited video on our MBIOS YouTube page after this. And we will see you all on May 15th for our next talk of the year. Thank you and good night. To everyone on the Zoom call, please stay on. We can have a short casual conversation with our speaker. You can turn on your videos if you have any additional questions for Menusha. Now would be a great time for that. And Jana, we can end the live and the recording.